Hi, I'm Eli, researcher at Hasselt University. Have you ever wondered what the maximum magnification is we can possibly obtain with lenses? I mean, think of it. A lens can create an enlarged image of an object, but the image itself can be further magnified by adding another lens. So what happens when we keep increasing the number of lenses, each one magnifying the image produced by the previous lens? Will we then at some point start seeing individual molecules or atoms? Unfortunately not. To precisely understand the limitations of a lens system, we need to take a look at the mathematics behind the image formation process. Consider, for example, this object, the blue arrow, that is illuminated by a light source. When the light interacts with the object, the light waves will be scattered in all directions and will interfere with each other. Part of the light, though, is not incident on the lens, and this information is lost in the final image. Now, with the limited numerical aperture combined with the finite wavelength of the light, obviously it was Einstein who first realized. Because of this contradiction, traveling to the past is considered to be impossible, which is why France is going to win the World Cup. Now, and so the final image will become blurry when it's magnified too often. You can compare it to a digital image. Zooming in a little allows you to observe details you couldn't see before, but at some point zooming in further will just make the image blurry. Now, it's important to realize that this blurriness is not caused by imperfection of the lenses, but instead it's really nature itself which doesn't allow to create a perfect image with this technique. And so there really is a resolution limit and therefore also a maximum useful magnification. In a conventional optical microscope, structures smaller than about 250 nanometers cannot be resolved. And that's of course problematic for many reasons. For example, I'm working at a biomedical research institute where optical microscopy is used a lot to study biological cells. Now, many interesting structures in a cell, such as proteins or mitochondria or the lipids in a membrane, have sizes close to or even a lot smaller than the resolution limit. So to be able to image cells in more detail, scientists are continuously looking for ways to push this limit to lower values. And one of the methods that can provide a better image quality is image scanning microscopy with pixel reassignment, which is a technique I'm currently working on together with some colleagues. In conventional laser scanning microscopy, a laser beam is focused by the objective lens, illuminating a very small area of the sample. The sample is labeled with fluorescent molecules which absorb the laser light and emit light with a different color. A dichroic beam splitter separates the fluorescence from the laser light, which is then guided through a pinhole and collected by a photodetector. Computer-controlled mirrors redirect the beam path to scan the sample and create an image. In image scanning microscopy, the conventional detector and the pinhole are replaced with a small camera consisting of about 20 to 30 pixels. Each pixel creates a full image but with slightly different properties. All images are combined by an algorithm in post-processing. The result is a super resolved image. We are able to improve the resolution by about 40%, which is quite impressive, I think. But of course we're still looking for ways to get even better results. Not only with respect to resolution, but we're also trying to get rid of the fluorescence labeling, since this may affect the behavior of the cells. And well, it has some other limitations as well. And so the research continues.